immense pleasure to introduce Dr. Prabhakaran Ponraj, the speaker of this session. Dr. Prabhakaran is presently the principal scientist in computational biology at Sanofi Genzyme, United States. He did his BSc and MSc Physics from Bharathidasan University. Later, he earned his PhD from Bharathidasan University for his research on X-ray crystallographic studies on some molecules of biological interest. After his PhD, he became a postdoctoral research fellow at the National Cancer Institute, National Institute of Health, United States. Later, he worked at the Duke University. He had been a scientist in several research laboratories of repute in the United States before he took up the present assignment. Dr. Prabhakaran has extensively studied the structure of antibodies, their crystal structure and sequences, especially those associated with HIV and SARS. He also developed databases and tools for genomics. He is an expert in antibody and protein engineering and has also developed several vaccine immunogens. Dr. Prabhakaran has published more than 50 research papers in reputed journals. He also serves as editor for several journals on immunology. With this brief introduction, may I now invite Dr. Prabhakaran to deliver his lecture. Over to Dr. Prabhakaran. Dr. Prabhakaran has to unmute, I believe.
Slides. Can you see it now? Hello?
Yeah, perfect. Yeah, perfect. Yes.
Can you see it?
Yes,
Okay.
yes.
Hello, everyone. Good morning. I'd like to thank Dr. Agastin and colleagues at Loyola College and friends for giving me the wonderful opportunity to present my work on SARS coronavirus. To begin with my talk, I had a statement to make. Actually, this talk was prepared and accomplished my own capacity, mostly the work based on the work I done during my tenure at NIH as a postdoc. So the opinion and ideas, suggestions, or anything do not reflect the view of my current employer, Sanofi. I just add thanks for introducing Dr. Joel. So my first postdoc actually after the Bharatdasan University, I went to Japan. I spent more than 3 years in the bioinformatics field at Riken Institute. At that time gave me a lot of opportunities to study biology and also structural chemistry, and it gave me a very good opportunity to equip myself before I move into NIH for a second postdoc to do protein X-ray crystallography. So I should thank yesterday's speaker, Dr. Saravanan, who gave us a very good introduction about infectious disease, particularly the vector-borne pathogens, and also he touched upon the introduction to SARS coronavirus. So today I'm going to talk entirely on my work on SARS coronavirus. Actually, you will be amazed. This work actually done in 2004 and 5, so exactly 15 years ago. So I'm presenting my own work, the crystal structure determination of a SARS coronavirus receptor binding domain in complex with antibody. Just for the information, the background in this title picture, the red colored tube you have seen, this is exactly the coronavirus spike protein, S1 domain. It's the top of the SARS virus. I am showing only the 2 amino acids. Those are hydrophobic and completely outside. That's probably used for the entry of virus. The background, the 2 colors, yellow and blue, are antibody. So this picture actually X-ray snapshot or X-ray photograph that the virus caught into the action, how the neutralized antibody neutralizes the virus. So here today, I just, as I mentioned that outline of talks, I briefly give the introduction about the SARS coronavirus, and then I mainly focus on my work on the SARS coronavirus, the structural biology work, and then I will move on to discuss what is the current status and what the structural biology of the current status, and then I will move on to discuss some of the key challenges and opportunities. So when I moved to NIH as a postdoc in 2002 October, immediately after a few months, the first SARS outbreak started. It's 2002 and 2003. These are the 3 major isolates, Urbani and TAR2 and Hong Kong isolates. The second outbreak actually happened in the following year. This is from the Guangdong GD03 isolates. And also we got some zoonotic transmission from palm civets. So these are the 2 more isolates. So given this background, 15 years ago also the virus posed a lot of threat to the humanity. So right now, this is compared to the previous is nothing though. So right now, even in the United States, we have a casualty more than 1 lakh. The whole, I mean, this is a kind of a major outbreak in the history following the 1918 pandemic. This novel coronavirus or the new coronavirus also, I'm going to refer to as SARS coronavirus 2. So today's topic that NIH at the lab, I had a very good opportunity to work in that NIH lab. Is one of the major labs in the United States. Actually, it's a cancer institute, but our group is an entirely virology group. So we quickly responded to the virus. 3 to 4 years, we spent a lot of time to work on this virus. So my major role in that lab is to first time we solved the X-ray structure of a neutralizing antibody in complex with SARS coronavirus. So the latter part, as I mentioned that, I will show you how this 15 years ago, the work we did make still impact on the current SARS coronavirus research, and then opportunities and challenges in the SARS coronavirus. I'll just quickly give you the overview. Yesterday also you probably heard about some of the things. This is the genomics, how the genomics looks like as compared to SARS coronavirus. So the coronavirus itself is 3 major families, alpha and beta and gamma coronaviruses. So the SARS coronavirus exactly belong to the beta coronavirus. So here the first one is the murine hepatitis virus genome. It's a reference genome. So there are 2/3 of the protein containing this non-structural protein the open reading frame 1 and open reading frame B. So these are this non-structural protein. The rest of the protein is very, very critical. The structural protein, if you look at this microscope, this is actually, this is a positive strand, single-stranded RNA virus, coronavirus family. So this is a rough, this is actually this diameter. When you look at the microscope, it's around 60 to 140 nanometer diameter, and each spike is about 12 nanometer. So the genome is exactly about 30 kilobases. So the protein, the whole protein has 10 proteins. So the transmission is very obvious. It's animal to human and human to human. So this, as I mentioned, that the 2/3 of the proteins are non-structural proteins. These proteins produce all the machineries required for the replication and transcription, and also the proteases, major proteases. So those are this, they make the viral particles. Then other the structural protein, those are the spike protein. This is a very, very key protein because the spike protein actually involves into the binding to its receptor ACE2. Here is the list of different coronaviruses and different receptors. So this is a angiotensin converting enzyme 2. This enzyme is very, very old enzyme, ACE enzyme. Those are involved in heart function. It's a very well-known enzyme, very old enzyme, but it turns out to be the SARS coronavirus. Even 15 years ago, and also there's a new virus, they both use the same coronavirus, the receptor. That's both coronaviruses use the same receptor. So in the next slide, I will show you the life cycle of the virus, just to recap. And then I will show you that on the right-hand side, what is the strategy that our work. So the left side, this is the life cycle of the virus. The very first step is a viral entry. The SARS coronaviruses use the spike protein to attach to the receptor ACE2. And then that fusion occurs. The conformational changes occurs in the S1, S2 domain that leads to the fusion of the protein, endocytosis, get inside the virus. That RNA genome immediately got translated into, as I mentioned, that there's a lot of non-structural proteins that makes this machinery to make the proteins, and also the replicates of the RNA enzyme get inside. So here is how this the whole inside the human, the virus make it possible to make a lot of viruses inside. So this involves also is hijacking the cell mechanism of, for example, ER and Golgi, everything it uses, and then it's a new virus budding out. So, for example, since this is a major epidemic now, there are a lot of ways to overcome the virus. For example, you can here the whole mechanism is going. For example, each protein is producing, and then the protease has to cut the enzyme to make the new particles. And then it's a lot of here the virus budding. Also, it uses a lot of host factors. So these are the targets for each and every step. There's opportunity to target to block the virus. But if you look at it here, this is here. I'm now going to show you here the right side, the receptor binding domain. This is the very first opportunity. You can block the entry of the ACE2 itself. Thereby, you can just block the virus. Then no more virus gets inside. Only you have this, only the nucleus. So here, this is a SARS coronavirus. This is a S protein, and this is antibody. This is a Y-shaped antibody, right? So this is an antigen binding fragments here. This is a 1 Fab, antigen binding fragment 1, antigen binding fragment 1. This FC portion is binding to cell. So this is what our lab, because as I mentioned that we just started to the SARS virus research. So we do not know, we don't have any clues how's the structure of the spikes itself, because there is no structure at that time. When I say the structure, the 3-dimensional atomic coordinates of the virus particle, of the each atom in the S protein. Because in order to understand the function, and also in order to understand the interaction between the ACE2 and other proteins, we need the fundamentally structure. So our work, we have to express the S protein, and also we have to make a perfect antibody that binds to that, and then we have to use the technique called X-ray crystallography to determine the structure. So this is the very first work we did. So this is my boss, Dimitro. So actually, as I mentioned that we were in the kind of race because we are in the very few labs, and also we know that Harvard University and a few others in different places in worldwide. They were competing for how to express the ACE2 molecule and also the SARS S protein. And then everybody wanted to solve the structure to find out the mechanism and everything. So we were in a kind of race. So our lab narrowly missed the discovery of ACE2. Dr. Michael Farson at the time, who was a professor in Harvard, he actually discovered ACE2 as a functional receptor. So in science, this always happens. There's a lot of competition. Then the Cell Journal asked our boss to write a preview about this paper. So the title is very catchy, The Secret Life of ACE2 as a Receptor of SARS coronavirus. So immediately my boss asked me to do that. What he wanted to do is, since we know the receptor now, why don't you just make a model of the ACE2 and also probably you can try the model of the spike protein. So that's what I made a ACE2 structured model as a SARS-CoV receptor. This is a paper that since that SARS virus is new that time, 15 years, they immediately put as a cover article. So in that article, what I did is I made a nice model of the ACE2 structure, and also I made a lot of different models for SARS coronavirus protein. So this model actually helped us to do a lot of lab work, wet lab work. Actually, this is a first step, as I mentioned, that we need to make a SARS protein domain. We have to find where the receptor binding domain happens, because as I mentioned, that 30 kilobase the whole genome, but the SARS protein itself, the spike protein itself, is about 1,200 amino acid residue. It's a very big protein. So we have to, that the question was the first, where exactly the receptor binding domain resides and which domain is a functional, because we have to find the functional domain as well as the receptor binding domain. So the paper just I showed you the previous, that helped a lot of variants, a lot of different lengths of the S protein we characterized. So this protein actually expressed in the one of the expression system called the baculovirus. So it's a kind of a difficult to work with. It's like a yeast because it can produce glycosylation. So these people are very, very expert, the top-notch scientists. They could express a lot of different forms, and then they finally localized where the RBD located in between the amino acid 303 and 531. And then actually they actually this protein, having this protein with a high purity, it gave us the opportunity to study the structure. Then this is the one part of the research. So this work was done by our molecular biologist. At the same time, there's another guy who is Dr. Zhu. He's a very good phage display technology. Phage display, this is one of the technology, right? To get the antibody, there are a classic method called hybridoma technology. You take the mice and immunize the mice and then get the antibody. So that's a hybridoma technology. That's a very well established. It's already got a Nobel Prize several decades ago. But this phage display technology also is a kind of in vitro technology. So 2018 Nobel Prize was recognized for this technology. Went to Professor Craig Venter and Francis Arnold. The Craig Venter is an antibody guy who actually contributed. So here how this phage display technology, they created the library. They collected the B cells from 10 healthy people, and then they make a library. So library of heavy and light chain, they collected from the B cell, the DNA, then they make a kind of, they make all this to make a vector, and then they put this cDNA inside the phage particles, right? So the phage display is a process of panning. Once the phage particle express the antibody, they will pan the antigen. They will, this is a SARS virus, the RBD domain. So they will select which RBD domain binds to the antibody. Then they will keep on the panning rounds, different cycles. Ultimately they will express the antibody with a high affinity antibody. So this is a IgM. There's a different antibody format. This one is a very, very naive. So this is how we ended up with the discover that our group. You can note that this is how this SARS research is now also doing. Even though it is 15 years ago, the same protocol is applied now. The people are doing the same thing. So first we have to have this S protein, and then we have to have the new antibody to make a complex. So here, this is you can see that how these people are very, very working day and night and make the very purified protein. So this Fab here is a Fab, and then this S protein fragments about 35 kilodalton. It's not that much big protein. So this is a Fab. So this is a binding experiments. how this binding affinity between this RBD and Fab and also RBD, the whole antibody domain, how it looks like. This is a surface plasmon resonance, the physical experiment to show how this binding the K on K off, right? So we can KD value can be measured. It's about 33 different concentration with a very high affinity antibody. So finally, having the good protein of RBD domain and then antibody structure, then the people as just I was introduced as a physicist. This is the first time I mean in my life that I went to the wet lab and then they gave me that protein and also antibody. Then my work was started. How to the very first step in the X-ray crystallography is to make the crystals, right? So I took the two protein. This is a very, as I mentioned that we were in the very tensed situation and we were kind of a rush and a race. Who will find the first structure? I saw that my samples, this is a very few milligram, even it's very few milligram. One of the method we have to do is we have to make a complex and then we have to run gel purification and then we have to purify the complex. But I cut all the steps because I know that if I do all those process, I will lose a lot of proteins and cannot get any protein. So I kind of I know that since I see since I already saw that protein is already pure, I just mix the two proteins and then I incubate over the night on the refrigerator. And the next morning I came and then make this kind of a crystallization. If you look at this one, this is how the protein crystallized. We took a small microplate. It's use the robo to mix the two protein, right? And then we put, this is a 0.2 microliter. Can you imagine that? It's a robo. So we make each plate of 96 well plates, right? So at a time we make 5 or 6 plates. It's a robotic screen. It will go on pipetting 0.2 microliter mixed with a SARS envelope and the S protein and antibody. And then it will add certain chemicals. This is a key. This is called a buffer solution. So we bought this buffer solution from companies. Those are matrix kind of a pre-mixed solutions. So this crystallization is actually, it's not a science. People say that it's a kind of art, because what I'm saying is we cannot predict based on our physical or chemical properties. Probably you can based on the pH and the precipitate, you can have some kind of initial conditions, but it's very rare to get exact condition. So after a 4 or 6 months trial, and I go back to the team and then I will say, I need to cut the sugars, because the sugars, if you have too much carbohydrates in the protein, the folding and the process, it doesn't crystallize well. So they have to use the pentase enzyme to remove all the carbohydrates as much as they can. And then they give me a very nice protein. After the 4 or 5 months, one day when I went to lab and then when I look at the microscope, I got very excited because I got this kind of crystals. So this is how this crystal looks like. Then after, later on, I changed the ratio. It's a protein 1% and this 4% of the chemical. Then you can see the nice dragon. This looks like a dragon fly's wings, right? It's a kind of a plates. If you look at that, you will see the beautiful crystals like that. So this is the first step. This is actually a bottleneck in any crystallography experiment. So many structures are solved, but a lot of important structures we never solved because they never crystallize. So the crystallization is a very technique. Now there's another technique is coming up now for the last several years called electron microscopy. That techniques don't need crystals. Okay, this is the next step in the research, how to solve the X-ray crystal structure. I'll just give you the overall view, even though it's very technical, just for the sake of your interest, this is a kind of a personal account, how I solve the structure. So I will share I think I could feel the same when I solved the structure 15 years ago. So this is actually this crystal, I took the crystal to we have the experiment called X-ray diffractometer at home. That means inside our lab, but that crystal didn't diffract because the power of the diffractometer is very that home is very limited. So we have to take the crystal to this advanced photon source. Probably some of you know that when you study the physics, you studied about the synchrotron radiation, right? So this is a very huge, about more than 1 kilometer radius, the circular ring. So the high energy beam that the electrons were traveling very high density, very high velocity based on the magnets. Then this is around the place, there are a lot of beam lines. There's a lot of, so one of the beam lines here, so that the electron will eject it out, and then you put your samples here, it will bombard the electron, and then behind there is a plate, X-ray plate. That plate will produce this kind of image. So first thing, you do not know that your sample is correct and your sample has a protein, because sometimes you don't have any protein, or maybe it's maybe only antibody or only SARS. So it's very you can say that it's very thrilling moment to know what is inside. So I went to that beamline. Actually, 3 of our friends, we went, and then we collected we carried a lot of samples. One of the samples is mine. We also brought the SARS complex protein crystal in the frozen cylinders. Then when we saw that this kind of a pattern, I was very excited, because this kind of a pattern is information that you have the crystal that can be solved. I mean, that can be the raw data. It's real. So here, how it's solved? This is called the reciprocal space. This is actually, when you do that, you probably saw that a lot of X-ray pictures, right? This is how you picture the X-ray using the X-ray beam, the synchrotron beam. The protein molecules looks like. You see only the dots here, right? Actually, this has a 3-dimensional periodicity, so you need strong mathematics. Actually, you don't need to do that's a high-end programs. They use called Fourier series. This is called this is an electron density equation. This is a kind of Fourier series equation. So it goes like a cos, it's a e power. This is a sine and cos theta. So it's kind of a periodic representation. So it has to translate to the reciprocal lattice. So the key is that X-ray crystallography techniques, the bottom line is you need to make your molecules to arrange in an order, in a 3-dimensional order, certain symmetry. So mathematically, they are very, very restricted, and then they are predetermined. So that is one of the requirement you need. So once you get the data, raw data, and then once you do the mathematical calculation using the software, you will end up with this kind of cloud called the electron density. Then this equation actually calculates the number of electrons. So the program, when you learn to use the program, you can build beautiful model. For example, here you look at this histidine here, and then here this is arginine here. So you can go to atomic details. It's a 10 power minus 10. It's a 0.1 nanometer. So if you build, that's a program, very, very powerful program. You can build the model for you. There's a lot of techniques here. I just here this antibody. The antibody structure looks same. So this is called molecular replacement method. We can use to solve the structure. So here is how this is the statistics. I just want to say that the protein structure, the people look at R value. It's a kind of a residual error. So this value should be below 10, below 20 normally. That means 20% of error is acceptable. So this is the 15 and between the 15 and 20 is a well-resolved structure. And also it talks about the resolution, how the structure resolution. For example, 2 angstrom resolution is very good. 4 or 5, sometimes okay. So you have to be able to see your side chain and everything so that you can define the active binding site or the interaction site. So the next picture, I'm going to show the snapshot. So here is the moment we were waiting for, how this, as you mentioned that, I told you that this is a microscopic picture, right? The picture, how it looks like. So this is the real crystal structure. So this is the snapshot of X-ray photograph of the virus caught in action to bound to the neutralizing antibody. So this antibody we named as M396. So this antibody, as I mentioned, there is a 2-chain, right? This longer chain is a heavy chain, the short one is a light chain. So this is a VL, variable light chain. This is a VH, variable heavy chain. Actually, this antibody is mainly interacting with only the heavy chain. The light chain is very less. Here, actually this, when I show you the very first, my talk title, I showed you the red color tube. Also here, this portion is here actually. So this is the whole S1 domain. It's about 300 residues. This is called the receptor binding domain. I'm here showing you the receptor binding domain of the S1 domain that interacts with the antibody. So the resolution is 2.3 angstrom. That means you can distinguish, you can go down to that level. So if you look at the picture, you can easily figure it out which part of the S protein makes the binding to antibody. So that way you can make I told you that antibodies already is a very good affinity. So this antibody can be used as a therapeutics in human. I will talk about a little bit very quickly in a few slides. Then other way, that other important information is now you know the real structure of spike protein. Then now you know this is binding to antibody. So in immunology, you have some clues to make a vaccine construct. If you use this construct as a vaccine, for example, this part or the whole domain, it can create an immune response that can generate antibodies, and then the antibody could bind like this. So this is a twofold benefits. When you solve the antibody neutralizing antibody, the first major thing, you found the neutralization mechanism. So you find where the antibody binds in the spike protein, and then you will see this. I think actually the another application is, you know that I told you, right, this resolution is atomic resolution. So you know the single residue, where is the single residue located? So you know the function of this spike protein, which residue is involved in the binding and which residue. This will be very, very informative information when you do that mutagenesis study to improve the antigenicity of the target to make a vaccine, or you can even improve the binding affinity. For example, if you take this antibody binding region here and then you can mutate and you can do more bench work to make the antibody like this. I told you that this is a micromolar, it's a nanomolar affinity. You can improve to the picomolar affinity. So this antibody, when I saw the structure, we got very excited because this is a first human antibody, neutralizing antibody structure with SARS-CoV. We saw in 2004, actually in December. That time I was heading to India for vacation, so I was very much worried that I would write the paper very quick and submit it to. So we submitted within a few weeks. I think I refined the structure, and then we wrote the paper and submitted to Nature, and then Nature immediately sent for review because that was very, very first structure was sent for review. So I had within 3 or 4 weeks, we got 3 major reviews, and the reviewers agreed that the structure is very informative and it's a very first structure. But getting to Nature publication, sometimes it's very tricky in the sense that I think I will show you that reason why. Then we resubmitted the paper to Journal of Biological Chemistry, which was immediately accepted within a week. By doing this structure, I know that this structure is going to be a gold mine because solving the X-ray crystal structure in the past, for example, you know that first crystal structure of DNA, James Watson, Crick, they got the Nobel Prize. And then if you look at this, all of the biological Nobel Prize in the first 20 or 25, even the very recent Ramakrishnan, he solved the structure of big complexes of ribosome, and also the previous structures, the very first structures, Kendro, the hemoglobin and myoglobin structures, and each of the biological macromolecules, like Dorothy Hodgkin solved the structure of vitamin B12 and the insulin structures. So these structures are kind of gold eggs hatching the duck. So once you solve the structure, the olden days, you can easily get a Nobel Prize. And nowadays, if you get the structure, very hard structure for the HIV and SARS or Zika, dengue, it's definitely go to the high impact journal. Not only the publication, but also this leads to the scaffold for designing the vaccine or antibody. So I will go through the very quick and end up with what is the work we have done. And then the next few slides, I will dwell upon how this, you will be surprised how this structure is now making the impact. And then I will I will discuss with some of the importance of this work, and particularly the challenge, or work of the challenge, and then I will open up the questions, and it will be very useful. So here is this is how this X-ray crystallography technique. It's a very high reliable technique, right? So it shows you that electron density, for example, if you look at this tyrosine, you can clearly see how these electrons are surrounded by the tyrosine. Then only you can put this tyrosine side chain here. So this is a major difference between the prediction and modeling. So prediction modeling you do, but nowadays it's becoming very, very competitive. That modeling also is very accurate. So once the structure done, you have to have a kind of theoretical verification, experimental verification. One of the experimental verification is Dr. G.N. Ramachandran plot. So you submit your structure to Ramachandran plot, and a lot of pre-check program that will check your structure for the high stereochemical accuracy. So here I just wanted to say that when you study the antibody complex structure, not only for SARS, HIV, and any infectious diseases, you have to look for the loops. For example, H1 is heavy chain CDR. CDR means complementarity determining region. Actually, when you see the antibody structure, the whole antibody looks like Y-shape, right? But if you look at this, the tip of the antibodies, those are the hypervariable loops. So here, H1, H2, H3, 3 loops in the heavy chain and 3 loops in light chain. They are highly variable. They actually adopt different shapes specific for different antigens, whether it is SARS or HIV, dengue, or whatever target it is. So here, these are the H1, H2, and H3. They are major contributors from the heavy chain, and one of the light chain 3. So this green color are called the epitope. These are the residue or structural fragments from the SARS coronavirus S1 domain, spike domain. So this is just to give you the glimpse of how these atomic interactions. You can see the water molecules. Actually, this is very, very important points that even though the virus recognizes antibodies, sometimes you see sometimes there's not much shape complementarity there. So what happens is nature puts some water molecule inside and then make perfect hydrogen bonds to make a very good complementarity, chemical complementarity, and also the structural complementarity. So this is how the structure should be done, and so this is a very classic example. So this is how this structure. This is like, you can imagine, this is like a lock and key, right? So this is S protein, spike protein. This is antibody. So this called the spike protein binding site called epitope. This antibody is called paratope. This is highly structured. If you put this on top of that, this yellow part will go here, fit in this green, right? So these are the how big the surface area for the interface, 800 angstrom square. This is roughly the same for most of the antibody antigen complexes, roughly 100 each. Yeah. So this our research finally pinpointed the paratope on the antibody and also the epitope on the SARS coronavirus. So here is why our paper was competing in Nature and couldn't make up is, as I said that the Harvard University also, they were involved in the study, but what they found is they actually solved the ACE2 complex, the receptor complex structure with the S1 protein. So it is very exactly complementarity work. I do not know how the Nature is missed our paper. Sometimes it's happened that this is the ACE2 molecule. Here what I did is I superimposed both of our complex, the complex from Steve Harrison from Harvard University. They solved the X-ray crystal structure of SARS S1 protein, RBD domain in complex with ACE2. This blue or cyan color here. The yellow color complex is mine. The antibody is our antibody complex structure with this S1 domain. If you look at that, the both structures, the S protein is very, very highly conserved. Here, if you look at the structure, I'm going to show you the next cartoon, but here you can see that highly overlapping, right? The antibody is overlapped. It's kind of giving you steric hindrance between that ACE2. So now this, comparing the two structures gave you the mechanism. How this antibody works is simply by interfering with ACE2. So our antibody now makes the point that it actually inhibits the ACE2 by competing with the ACE2. That's the neutralization mechanism. If you look at that here, this circle is a common space that antibody binds here, where this receptor binds here. So this is the overlapping epitope. So here is the cartoon, we just made it, how this RBD domain. Within the RBD domain, there's a portion called receptor binding site. So this receptor binding site is a purple triangle. So that's where this antibody and ACE2 binds. This is actually the receptor binding site. These residues are foothold or kind of gate entry for the ACE2. If you have a perfect antibody, it can outcompete the ACE2, and then you can just block the virus. So also we also find that no change in RBD, because if you look at the structure from the ACE2 and the antibody complex, it looks same. So we hypothesized and we concluded that there's no conformational change, even though the ACE2 and between these two conformation changes. So just going to quickly run through, because of this work is very, as I told you that we had a subsequent outbreak in this next year, 2004. So we had, we developed, I think it's a very huge collaboration with NIH and our group. So we developed animal models. So we tested the animal model, how it neutralized the virus. Actually, this is the data. How this neutralizing data, it neutralized the virus in animal model. And also I made this bioinformatic analysis This is at that time I collected all the sequences from different SARS coronavirus isolates from different regions, and then I found that those in the red color, they are the antibody binding sites, they don't have any variation. On the other hand, the blue, those are the escape mutation. The point here is the antibody focus on the conserved region, not the hypermutation region. That means the antibody is able to cross neutralize, or it's a highly conserved target. So this is a point. The red color is antibody binding. The blue is escape mutation. So only one escape mutation actually overlapped, but we saw that it doesn't make any difference whether it is tyrosine or serine. So we hypothesize that the molecular mechanism, the cross reactivity, our antibody has a better cross reactivity. So with that, I just conclude that part of the main part of my talk, how this we applied X-ray crystallography 15 years ago. Now I'm going to surprise you how these results are now making impacts in the SARS coronavirus research. So the conclusion is now, as you see, that we actually provided the very first time the structural basis for the potent antibody neutralization for SARS RBD using X-ray crystallography. So as I mentioned that the structural knowledge can be guide vaccine design, and also the 396 antibody itself can be a new therapeutic applications. We can develop powerful antibodies. We can use this antibody, or we can make powerful variants antibody. So having seen all these results, actually that less than a year ago, when the SARS outbreak begins, the novel coronavirus came in 2019, I got a mixed feeling that I thought that the M39.6 can be the key, can be the very powerful drug for the new virus, because I know that this virus, the antibody targets a very conserved region in the receptor binding domain. Also, the new virus also, I want to remind you that the new virus also bind to ACE2. So this antibody actually compete with ACE2. So I thought our antibody is going to it will be a game changer as a therapeutic antibody use. So I thought it should bind, but what happens? I'm going to show you. So that is one of my NCI boss, Dr. Jinwaji. So he has a very good crystallographic group, and he provided me, as I mentioned, that my other NCI mentor is Dr. Dimitro. He's a virologist. So I work for the two group. So actually, he put the news recently that in JBC, Journal of Biological Chemistry, they published the coronavirus issue, that free issue collecting the high impact journals published so far. It's not only one antibody. Later on, other people also, I think 3 or 4 antibodies complex structures were solved within a year or two. So all this compilation they publish here. So this actually still in NCI website. I think they put 2 or 3 months back that how the structure was added into that. So here I'm going to show you, this is a first Nature paper. This is a first Nature paper in 2020, a few months ago. They reported the new SARS coronavirus structure. So immediately the first thing they did, they compared the structure of SARS coronavirus, RBD. Actually, they also saw the RBD, same structure, but with the ACE2 enzyme. This is a new virus, but the old enzyme, right? This is ACE2 same. So here is the sequence comparison and structural comparison. If you look at the structural comparison, this right side is the old virus, SARS coronavirus. You can see 1, or it's the old virus. This is a new virus, coronavirus 2. Look at that. This top portion is the RBD S1 domain, protein S1 domain, receptor binding domain. They showed the red color stick amino acid residue. Those are highly conserved. Look at that here, tyrosine. And if you look at that histidine, I think this is the histidine, right? But I think that's what they found is the most of the residues between the SARS and the structurally conserved. These are the ACE2 binding residue. ACE2 binding also highly conserved. For example, here tyrosine and histidine. And the point of this Nature paper is the new virus RBD binding domain and ACE2 interactions are highly conserved. Only here, if you see that RBD contacting residue and RBD contacting residue in old virus, coronavirus, this is a coronavirus 2 is a new virus. So if you look at here, they are same. For example, here, their binding site, and some of them, they are same. Some of them are different. That's why I mean that, for example, the new virus and old virus, the structural binding site for ACE2 are same, but still it's very amazing that how this is different. You can argue that for example, the blue color, those are highly conserved residues, right? Those red residues are different, right? Even though there are quite notable differences, the virus is able to recognize the ACE2. That's amazing. Actually, this paper is actually cited our paper. I'll show you that another good example. So here's the first thing. As I mentioned that, as I was thinking when the SARS outbreak came, I told you that I immediately came to mind that M396 may be useful for neutralizing the virus. So this is one of my previous colleague who is now working in Fudan University in China as a full professor. He immediately tested the virus. Actually, he has a clone for this virus, the sequence. So he cloned the antibody, and then he checked. He found that this antibody doesn't bind to the new virus. Look at that. But only one antibody. This is another antibody isolated. It's reported also in that same time. I mean, 10 or 15 years ago. Only this antibody binds. There's another group. So here, this is a new science structure. This is also citing our paper. So here, that if you see the title, so here I'm putting the two major citations, Science and Nature, that cites our paper. So the point of this science paper is that's a new virus. They found a new antibody. Actually, our antibody binds right here. This is the ACE2 binding site is right here, almost here. But this antibody binding is in a weird position. I mean, this is a weird site, I can say that, because if you look at that, this is only the fraction of the S protein, right? Actually, the S protein has another S1 domain. Actually, then that virus actually is a trimer, right? So you can imagine trimer. Now the new structures came in using the electron microscope that has a whole structure. So if you imagine that another S protein here, I do not know how this antibody is going inside. Probably there may be some mechanism. But also this antibody, they are not sure about how it's effective because it's not binding at the place of ACE2, and also they don't have any efficacy data. But still they published that science because it's the first structure of new virus and antibody. So here is another paper. If you look at that, this is another Nature paper. Here they actually cited they actually dedicated one picture of this M396 antibody. This is another antibody, 18R. So they compared the two antibodies from the first outbreak 15 years ago. So they compared the new and the old and new virus, coronavirus 2 and coronavirus, and then they mapped the binding set of antibodies. And then the point of the paper is they say that the M396 has only 7 change, whereas the 18R has 20 changes. So they conclude that M96 is very, very likely to cross-react. It's not a cross-reacting, but they are saying that's only have a very few mutation difference. So what does it mean is this paper, after seeing this paper, me and I think everybody in the field thinks that this M396 antibodies can be re-engineered to bind to the new virus. So that is the point, take-home message, right, from this previous paper and then this Nature paper. Now we know that I didn't show you here, but if you look at, I think I showed you here. Look at the two structures are same, right? So our antibody binds to this old virus. But here there are a few changes, though immediately the hypothesis is we can change this antibody a little bit. We can tinkering the antibody, that's called the antibody engineering, make it bind to here, right? That's the point of this paper. So here, as I mentioned that the paper, actually this is a conclusion from the paper, Nature paper. The M396 has only 7 residue changes in SARS-RBD among 21 epitope position. Look at that. The total, this is only 1/3, right? Exactly 1/3 only changes. So 2/3 are conserved. So it suggests that clearly this antibody can be powerful. It can be re-engineered, right? So this is what actually happened. That value is very small, and then science is very highly competitive, right? So this is a big national lab. The United States, it's like our CSR lab in India. There is we have certain labs called the national lab. This is called Lawrence Livermore Lab in California. This group actually used this is a physics group. Actually, they use supercomputing. Machine learning is a method to use our antibody. They made about 9,000 variants based on free energy calculation and prediction. Sorry. So they wanted to make this 396 antibody reshaping the specificity to new virus. So actually, this company actually gave I heard that when you read this paper, they're saying that they're waiting for the functional data. So I may not be surprised if they make it antibody binding to the new virus. This is the last one for the impacts of in this cell paper, they isolated antibodies from 6 patients. So this is one part of the figure. I'm sorry. Actually, this is one of the key figure. They put a structure of our antibody reproduced in the cell paper. So the major aim, I'm going to show you this picture actually I made. So 60 people, this is a zero conversion. Those are recovered patient B cells antibodies, isolated. And then what they did is, they took our antibody, the structure, part of the structure, CDR3 loop. Then they computationally predict the structure of CDR-H3 for each antibody. Then they use our antibody as a bait. Whichever antibody has the similar fragment compared to they consider only the CDR-H3, the prominent loop. Wherever the antibody looks the same, they take the antibody and then they clone and express the antibody and test. And then amazingly, the cell paper reported 14 antibodies. 11 antibodies they designed this way using this M39 series structure. It's I was very surprised how this antibody made an impact to make this kind of prediction. I think in the future, I'm thinking that this is going to be the very, very giving you that idea how you can know antibodies, and then if you have a similar targets, probably you can re-engineer this antibody. I mean, you can select the antibody. For example, the traditional way, if you wanted to do the antibody isolation, you have to go through a lot of procedure. For example, you have to this is actually B cell isolation, right? So once you collect the cell, you got to do proper screening through experimental. It takes 1 to 2 months, but if you do this bioinformatics method, so just a matter of days or a week, you can select this antibody. Okay, and this is impact. As you see that the whole work we did, our group, I think I can say that it's a kind of a very, very huge task done by different people in the group, like the molecular biologist and phage display scientist and X-ray crystallography by myself, mentoring by the NIH professor. And then this work is highly how they are, so far I am showing that this paper getting cited in Nature, Science and Cell top journals because of the reason how this antibody, the people are thinking that this antibody can be re-engineered or can be used to select the new antibodies. So here I'm just having a few slides before going to that QA or that conclusion. So this is the current status. So this antibody currently, as you know, the chloromethoxy, the malaria drug, and also the another Ebola drug, remdesivir, the remdesivir and hydroxychloroquine, these two drugs, not only the two drugs, there are a lot of drugs people are trying to repurpose the drug. Just some of them are FDA approved and some of them are very well-known drugs. So they wanted to see whether if you give the patients, it will have any clinical effects or not compared to the placebo. And also there's another, this is one approach, it's a crude approach, I can say the brute force. The another one is antibodies. As I just talked to you about 40 or 50 minutes, maybe about 55 minutes. So antibodies you can recreate using the mouse with a human immunoglobulin or even the mouse. And also you can, I just showed the last slide, cell paper. We can also get plasma B cell from the PBMC collect from this recovered patient. You can make antibodies, and also you can also bioengineer. I just showed you the Lawrence Livermore lab. They use a supercomputer. You can design antibodies. So these are very, very, I think this is going to be making a huge product for biotechnology. If you have a very good antibody, it can definitely succeed in clinical trial. For example, one of the antibody we have RSV, right? RSV virus, and also it's a lot of clinical trials are going on. So it's another thing is vaccine. There's a lot of vaccine, RNA, DNA vaccine, subunit vaccine. This is the ultimate goal, right? So once you get the right combination of S protein and also powerful adjuvant, our company also Sanofi past year also worked with AstraZeneca, and so many companies are now doing this. Another RNA vaccine. Moderna, right? This is a big company. They are also working this RNA vaccine. I think it may take time, a little bit time, but eventually we'll get it. So this is another way. I told you that this is a kind of brute force repurpose. Also, it's another science paper. They actually solved the main protease from this SARS novel coronavirus. So far, I showed you the structure of spike protein, but also, as I mentioned, that there are a lot of non-structural proteins, including a lot of enzymes and other accessory proteins. This is one of the main proteases. So they solve the structure, and then they designed some of these molecules, a-ketoamide inhibitor, the complex. So this may be that starting point to make this inhibitor to block the function of the protease, then that virus particle cannot be made inside the cell. then you can reduce the viral load, and then this is another option. So this is one of the final slide. I think we need to have a fundamental, very strong immunology and virology background to understand the chronology of events during SARS coronavirus infection. Actually, this review I got from Nature Immunology. So this title is very catchy. Like this title is the Trinity of Immunity, and there are three concepts: immunity and inflammation and intervention. This is called the Trinity of SARS coronavirus infection. So this is just I explained to you how this life cycle, and this is how once the virus gets inside the cell, and then it mounts a lot of response, right? T cell response and everything. So we have to understand that how the cytokines, a lot of storm, right? A lot of people are dying of this very dramatically T cell response, right? So we should understand how it works, and then we can make actually some drug targets here to clear up the very powerful cytotoxic cytokines we can remove from the system. And also another option is here also. There are another options here when you target the the macrophages and other stuff. So what this review is emphasizes that this explains everything, infection, and then knowing the more, if you know more about this thing, probably can have the better another opportunity apart from this. The three things I showed you, Bo, you can also make another thing. This is the last one. I think this is just probably for the sake of advanced information, I'm telling you this is a ADE. There is a huge, for example, everybody knows that it's antibody or any drug. There is an immune response called anti-drug response, anti-antibody, that your body, whenever you put something inside your body, that body will immediately mount the system called anti-drug response, right? You can create antibodies or T cells that will neutralize the drug. Similarly, there is another mechanism called antibody-dependent enhancement. What it does it, instead of neutralizing, blocking virus, this antibody, sometimes what happens is, it's a very loosely bound antibody, it's not that much tight. What it can do is, instead of neutralizing the virus, it can just put the antibody inside the cell, and then I think it's the problem is that's some binding sites. It can create the macrophages, infect the macrophages. Instead of clearing the virus itself, it lead to the enhancement of the virus. So this, you have to be careful about that. I think one of the SARS problem is people are speculating that this is ADE, maybe the culprit, the responsible is probably the big caveat culprit. I think this mechanism. So here also there's a lot of I think this how it's also there's a lot of thing, like as I told you, I never talk about the FC portion, the cell binding portion, the FC receptor. So it can also bind to the different the FC gamma receptor that can also lead to shift. This is for SRC domain. It's a tyrosine phosphorylation. I think this also giving you cell signaling. It's a negative regulation of immune response. It will shut down your immune response, and also some of these skewed macrophages and a lot of inflammatory cytokine. As I mentioned that we do not know much about these viruses also. For the large, the 2/3 of the protein, non-structural proteins, there are a lot of accessories protein. We simply do not know the function. Sometimes we have the structure, but but I think this hypothesis and the knowledge from the other viral diseases is some of the proteins they simply used for down-regulating the innate immune system. So that is also key. Whenever the virus is taking the advantage, once it gets inside the cell, it down-regulate some kind of, and also it creates the negative regulation as well as the inflammatory signal. It will just mask your immunity to this virus. So this is the whole thing. We have to avoid it. So final slides, the structural biology of SARS coronavirus advanced knowledge. So you can really actually appreciate that how this worked 15 years ago, and then now the current virus. You have I didn't show a couple of papers, the recent papers. They have this used the cryo-electron microscope to capture the whole virus, not only the S protein, but also the trimer structure of the S protein with ACU2. That gives some additional information. So it really gives actually, even though the SARS people are saying that we do not have any, even though the science is advanced, we do not know, but as you just present to you, we have a very advanced knowledge now about viral entry and antibody neutralization mechanism. And also the high throughput B cell sequencing from the patient sample, now it's very clear how this antibody can be very effective. And also the new vaccine development, as I mentioned that there's a different kind of vaccine, and also the viral vector methods, and then a virus-like particle, an inactivated virus to prevent the SARS virus infection. This is the ultimate need, what we need to prevent the next cycle of re-emergence of virus. We need to have this. as I mentioned, I want to end up with this major challenge still remain. For example, why this previous virus is easily contained within a few months, also within the China and other places. It doesn't turn out to be a major pandemic. The new virus, why it is too virulent, even though the structure and sequence are highly conserved. That is a key challenge to understand. And also the host factors, maybe the host factors may be different for the new virus, and also how this inflammatory response is handled by the ADE may be the problem. So finally, this work cannot be completed by my two big bosses. I mean, the two my NIH mentors, Dr. Dept. Now he is moved to Pennsylvania, Pittsburgh University, as he's now director there, and then he initiated the antibody center there. So he worked on a lot of, I think with him, I worked for last 15 years, I think, or 20 years, I can say. I still used to work with him. Our long-term major goal is HIV vaccine. I work with the Duke University, that's a big shot, and HIV field, Dr. Bart Haynes, and then he works with Bill Gates Foundation and so many things. And he has, I think it's over $1 billion projects. Last 14 years he's doing, and then actually we wrote. I think if you're interested, you can look at my recent publication and then a previous publication also that on the HIV. We wrote a very good article on 30 years. We reviewed how the HIV vaccine progressed, and also Dept, as I mentioned, that he worked on different viruses. Even Nipah viruses. Yesterday, Dr. Saran mentioned about Nipah viruses that are also very dangerous. I think in India also we have seen that cases, Nipah viruses in Kerala and some other places. So we developed already those antibodies also. I actually wrote a couple of articles and expert opinion about this Nipah antibodies and Nipah receptors and proteins. Dr. Jinwaji is a crystallographer, and then his major contribution is the crystallization of dicer. Is very, very advanced. I think this is CRISPR-related work. He did a lot of work. And then these are my colleagues, and they are very, very strong. Feng, their husband and wife team, they are very much a very major molecular biologist experts. And then Zoo is a phage display, and Samizab Chakrabarti is also a very key player. And then he also helped me in the crystallography with Chinua lab. With that, I would like to thank everyone for the listening, and I hope I can take some questions if you have. Thank you.
Dr. Ponraj, yeah. This is very nice and elaborative. We have quite a few questions from the participants. Shall I read it out? My voice is audible?
Yeah, I can hear it, yeah. I think I can answer some of the questions if I know the answers.
Yes, sir. Okay. Which expression system is for expression of ACE2 domain?
So I can hear you well. I think the audio is not breaking up.
Which expression system most suitable for expression of ACE2? My voice is clear?
Yeah, not. What is the question? I cannot hear you well.
Which system is most suitable for expression of ACE2?
Yeah.
Is it clear?
Yeah. Can you also, someone can type also? It's not clear. I don't know. I think it was very clear.
It's already in the chat box. It's already in the chat box.
Which expression system best for expression of ACE2?
Yes, sir. Yes, sir.
I think I remember that we didn't expect. I think as I showed you, we worked on the SARS RBD. This ACE2 was. I think our lab is expressed. Yeah, I have to go back and check it. I think it's a baculovirus again.
Okay, sir. Okay.
Baculovirus. Yeah.
Okay.
Because I can show you that ACE2 also, the glycosylation is very, very important for that virus. I think a lot of glycosylation. Even when I do the modeling, I consider the glycosylation and model the structure. So it should be, I think that suggestion for ACE2 expression system should be something like yeast or baculovirus. Yeah.
Okay. What is 2 and P in crystal conditions, sir? What is 2 and P in crystal conditions?
Yeah, I think I got to go back to that.
Okay.
I think I just gave the crystal condition. It's a protein crystal condition, so maybe I can share the slide. So 15 years work, right? I didn't have top in mind, but yeah, I can tell you that. Yeah, this is, I think, the crystal condition. What we got is a very standard crystal condition. I mean, it's highly reported. That one is produce a crystal. Later on, I had to play around the concentration, changing the concentration of protein and buffer solutions.
Okay.
I will get back to you later. Yeah.
Which crystallization method used in your study, either hanging drop or microbatch method?
I think it is a sitting drop.
Sitting drop.
Yeah.
Okay.
It's very old method. Yeah. 15 years sitting drop.
Question.
It's
Whether both protein concentration are same in the complex of crystallization?
yeah, whether both protein concentrations are the same in the complex crystallization. Yeah, I think that when you make the protein crystal, I think I remember that the protein crystallization, we always put at least 10 mix per milliliter, right? 10 mix per ml, right? So I think when you the protein crystallization, when you make a complex, I think it will changing, right? So it will. The question is, I think, the same concentration. Yeah.
Fab M396 is clinically acceptable.
Yeah, that's a good question. Yeah, I think, yeah, the point is a very important point. This antibody is a fully human antibody. So generally, it is expected that the fully human antibodies most likely will not have any adverse effect. So you should go through the clinical testing. If it is, I mean, whether the antibody, someone changed this antibody, re-engineered this antibody, or as you see that Cell paper, they have the same antibody, the similar antibody sharing the CDR-H3 sequences. Those antibodies actually produced from the human itself, right? From the patients, recovered patients. So it should be accepted by the, I think, I expect that this will not have any problem with the clinical acceptance, but still it has to go through the protocol.
Are the antibodies protective from COVID survivors?
Yeah, this Cell paper, I showed you that, right? They took the antibody isolated from 6 recovered patients, and then they showed the very promising data that it worked in the laboratory data. I mean that laboratory data, I think they have to have this clinical trial should be done. Then only we can use this antibody for clinics. But definitely it should be protective, because this antibody, they have a lot of assays to publish the paper to get on. They did a lot of assays comparative to other antibody, so it should be protective.
Why it's difficult to prepare vaccine for COVID-19?
I think it's not my field, but I think still, difficult to prepare for vaccine for COVID. I think if you look at the history, I think so many of the vaccine development takes time, but I think this one is, I think it's a new virus. As I mentioned that even though the structure looks similar, there are a lot of mutations, so you have to be very careful about which lineage has to consider, and that is the, I think it's a new virus, that's the problem. Yeah, it takes time.
What are the precautions you have taken when you're working with exactly infectious virus?
I think entry level also, I just want to point out, for example, the HIV, the company, very famous company called BMS. So they also developed some kind of small molecule drug that target to the GP120 for the HIV. So the similar way, the small molecules can also be tried to develop. I think people are already doing that. For example, I think the entry level, I think the replication level also very important, because I think the replication level also, I think, for example, that some of the protease drugs, for example, the HIV, once upon a time, it was like a death sentence, right? People will die very quickly in a few years, right? Now people are lifespan
a long back your work. This work are done in the long back, but it's all very good for us. Okay, from you have given very good information. Okay, definitely many people will take up this for their future work. So, though the subjects are a little bit difficult for us, you have given all the information very clearly, particularly the X-ray crystallography, data collection, structure prediction, like that. Okay, in most of the cases, you have given your own research work. That is very good. Okay, some other data and presenting. Okay, that shows you are confident in presentation, and you also given current strategy for the development of the vaccines for the cure of the COVID. Thank you very much, sir, and also thank you for your sparing your time, though it is midnight for you. Thank you very much. Thank you very much for answering all the questions by the audience. Thank you very much, Prabhakaran, sir.
Thanks, Dr. Agastin. Thanks. Thanks, everyone.
Thank you. Thank you. Counted for your e-certificate.
Dr. Prabhakaran, thank you so much for being with us.
You.